Our next speaker today is Sharon Gordon, and she will be telling us about the work from Flagship 2.
So good afternoon, everyone. Thank you for coming. I'm Sharon, and I fall under the Translational Theranostics Flagship. In this flagship, there's two researchers. There's me, the basic science researcher, and there's Dr. Vinu, the clinical side. So translational research is really the bridge between basic research and clinical trials, pointing the term benchtop to bedside. So just a brief background about theranostics. So theranostics basically can use any biological marker within the human body. But for my research, I'll be using the neuroendocrine system as an example. So here is a basic diagram of the neuroendocrine system, where you can see around the body there are several organs that contain neuroendocrine cells. The commonality between these cells is the somatostatin receptor. So in theranostics, we can utilize this commonality receptor and attach a somatostatin analog. From here, we can attach a diagnostic radioisotope such as gallium-68, where you can see on the left a common dotatate PET scan where we can diagnose the patient and then assess the metastatic disease. From here, we can attach a therapeutic isotope such as lutetium-177. And here you can see a basic structure of lutate, which contains the somatostatin analog and a therapeutic isotope. So at Royal North Shore, we have a patient protocol for lutate treatment. First, we work up the patient using, we assess their renal function, we assess the quality of life, and we assess the bloods. We then give them 4 cycles of lutate with an average interval of 8 weeks between each cycle. We then assess between each cycle their quality of life and their bloods. And at midpoint, we reassess the dotatate scan and their renal function. After this, we have an average of 3 months follow-up assessing all of the above, as you can see in this diagram here. So for my project, we did a retrospective single-center analysis of the safety outcomes of patients that underwent lutate treatment between an audit period of 2013 to 2018. We also assessed one of the dose-limiting organs in the cell, the kidneys, where we assessed the renal dose and if that has an acute impact on renal function 3 months post-treatment. We also wanted to see the baseline and mid-cycle renal scans and if that gave a clinical insight into a patient's post-treatment renal function. So here is, with our cohort, we found an average baseline of 70 mils per minute corrected to body surface area. This then dropped post-treatment to 75.4 mils. You can see here, this is a bulk plot where you can see we plotted the difference between a patient's post and pre-renal function against the average of the two. We found that there was an average difference of an average drop of 3.6, where the clinically significant is defined as average of 10%. So we did find that there was 39 patients that fell below the clinically significant decline, but this was only 3 months post-treatment. So you can see from this graph that majority of our patients were reasonably stable, centered around a 0%, zero change. Now we wanted to see the renal dose patients received during our treatment. We found that on average, patients received 10.6 gray across the 4 cycles, which falls below the accepted threshold of 20 gray for the kidneys. We then wanted to see if this renal dose had an impact on the renal function. So we split our cohort into 2, patients that had less than 10 gray and patients that had greater than 10 gray. We looked at the percentage change between the pre and post renal function, and we found that there was a drop of 2.4% for patients that had a lower than 10 gray dose, and then a drop of 3.8% for patients that had a higher than 10 gray dose. We found that this difference was not statistically significant between the two groups. So now we wanted to look at the impact, the contribution, and benefit. So one thing we looked at was the baseline renal function and if that had an impact on post-lutate or gave us a clinical insight. So here we found that when patients had a baseline of 70 mils per minute, the post-lutate, as you can see here, didn't fall below 50 mils post. So when we look at our patient protocol, we can see that we can safely omit a mid-cycle evaluation for these patients, saving them money and time, and also freeing up the scanner for other clinical needs. So if we look at our patient protocol, this does impact a significant amount of our percentage, where you can see nearly 30% of our patient protocol is from the inner regional areas and 8% in the outer regional areas. So this is saving them a lot of travel time going to our center to get the mid-cycle scan. And lastly, we looked at the mean overall survival and mean progression-free survival. So we compared this to the clinical trial NETA-1, where you can see in the graphs below, Well-Norshore was the red line and NETA-1 was the dotted blue line. You can see that our mean overall survival was 72.5 months, and our mean progression-free survival was 34.3 months. Where you can see at the cutoff for NETA-1, they estimated that the progression-free survival at 20 months was 65.2, where you can see at ours was 84%. Yeah, that's it.